Well, welcome back, everyone, from your break. I just wanted to remind everyone that the chat box is open. So if you have questions or comments during Dr. Tromberg's presentation or other presentations as part of council, feel free to use the chat box. So with that, I want to take this opportunity and thank and welcome Dr. Bruce Tromberg, who is the director of the National Institute of Biomedical Engineering and Bioengineering. In this capacity, he oversees a portfolio of research programs that are focused on developing, translating, commercializing engineering, physical science, and computational technologies in biology and medicine. So I've worked with him in his capacity as leading the NIBIB's Rapid Acceleration of Diagnostics program, the RADX Tech program. It's about $1.5 billion. And the focus was to increase SARS-CoV-2 testing. And in fact, I think he's just done a remarkable job. I mean, soon after we went into COVID lockdown, some of those tests that you could buy at Walgreens and CVS actually derived from the really innovative programs that Bruce brought the leadership to. So I think we're going to hear about some of those details here in just a few minutes. So prior to joining the NIH in January of 2019, he was a professor of biomedical engineering and surgery at the University of California in Irvine. During this time, he served as director of the Beckman Laser Institute and the medical clinic, the BLIMC, and the Laser Microbeam and Medical Program. So Dr. Tromberg received his undergraduate training at Vanderbilt and did his MS and PhD degrees in chemistry at the University of Tennessee, where he actually worked at the Oak Ridge National Laboratory. So I actually overlapped with Bruce at ORNL. We didn't know each other well then, but I'm not surprised that he's doing so well with that fabulous training at ORNL. So he has co-authored more than 450 publications, holds 24 patents, and has a long list of noteworthy awards. He has been remarkably collaborative. He's very interested in working with us at NIEHS to explore how we might utilize his background in bioengineering to create new technologies for collecting environmental exposure data and then integrating all this together in a way that we can really bring together exposomics and precision environmental health. So in the interest of time, without further ado, I'm going to turn the virtual platform over to Bruce, where he'll discuss his work in NIBIB. And the title of his talk is the NIBIB and the Pandemic Response, New Opportunities for Accelerating Innovation from In Vitro Diagnostics to Medical Imaging. So Bruce, thanks for being with us and take it away.
Thank you so much, Rick. Thank you for that very, very kind and thorough introduction. And hopefully I'll be able to cover all of these topics in this time and look forward to having a conversation throughout. Rick, I can't see the chat, so if you want to just interrupt me, if there's a question that comes up, please let me know.
We'll keep an eye on it, Bruce.
Terrific, thank you. So I'd first kind of like to introduce a little bit about NIBIB. It is the newest institute, and we're probably one of the smaller institutes as well. We were created in 2000 by an act of Congress, so all the institutes were created that way. It was a bit controversial, so the NIH director at the time, Harold Varmus, was opposed to it, as many were, because there wasn't really an understanding of the role that new technology development played in biology and medicine. People have always thought it's important, but there's been kind of a division in terms of philosophy as to whether or not technology development without a biological hypothesis should be supported within the NIH. So to underscore that, I have wording from the act that was, it was moved into public law by the 106th Congress at the end of December in 2000. And it really underscores in very kind of powerful words for the technology development community. It says basic research in imaging, bioengineering, computer science, informatics, and related fields is critical to improving healthcare, but it's different from the research on which the current NIH or the current institutes are based. So here's really the major imperative: to ensure the development of new techniques and technologies for the 21st century. These disciplines therefore require an identity and research home at the NIH that's independent of the existing institute structure. So as we sit around and talk about ARPA-H and its possible formation, I think we can go back to 2000, a little over 20 years ago, and recognize that there are these transformational times and events and opportunities. This was not widely supported, but ultimately it did win the day. And here we are as an institute. Now, what's happened since the creation of NIBIB, and with one particular foundation, the Whitaker Foundation, that's invested heavily in biomedical engineering, we've seen that human health has become a top priority in schools of engineering. If you go back to 2000, roughly $100 million in total was going to schools of engineering from all of the institutes and centers all across the NIH, an incredibly small market share. But what we've seen in schools of engineering since this time is more than a 5-fold increase in the formation of biomedical engineering departments. We have about 145 of them now throughout the country. The entire UC system is organized with a coordinated trans UC campus department, but every one of the individual campuses has departments. This is true for most of the universities, state universities, and leading universities around the country. We're adding around 1,600 new undergraduate biomedical engineering students a year. So it's a very, very popular major. So in addition to the students, of course, there are many new faculty that are being hired in universities all around the country. We've seen medicine and engineering partnerships emerge with kind of the formation of this notion of physicianeers. So the University of Illinois Urbana-Champaign has a medical school, the first one, where its foundational curricula embeds engineering. Texas A&M, the founding NIBIB director, Rod Pettigrew, moved to Texas A&M to start their first medical school with engineering embedded in the curriculum. So these are two foundational movements. We'll see this expand to medical schools even more throughout the country. And there are, I think, as many of you know, many centers for engineering and medicine or bioengineering with joint faculty appointments. I had a joint faculty appointment in surgery and in bioengineering in the School of Engineering, and I think this is becoming increasingly common. So all of these activities are doing they're having a very unique role, not only in advancing human health, but also in economic well-being, economic growth. Many of these centers, departments become centers or foci for innovation and entrepreneurial activity. And we're tapping into that with the growth of this entire field. So what are the topic areas that this represents? From it's bioengineering can be considered to be quite broad, but I'll try to define our vision and exactly the major areas that we support. Simply put, our vision is engineering the future of health. And we're concentrating our efforts in these major areas. One is in engineered biology. So you can think of this, I think, popularly as tissue engineering, but it goes beyond that. Some of the new trends and the new areas in engineered biology that we're continuing to advance are in looking at cells as models, and we can define them much like we can define integrated circuits. They have switches like integrated circuits do as transistors. Those transcription factors within cells can be viewed as switches in a complex cellular biologic system, and they can be reprogrammed. So much in the area of synthetic biology views cells and multicellular structures as reprogrammable, and that extends to the formation of tissues and extracellular matrix, the addition of vasculature. So this is, broadly speaking, the engineered biology area. We also support sensors and point of care technologies. Those sensors may be wearable, they may be implantable. They can be specific for particular molecular analytes or able to transduce physical phenomena. Point of care devices can be deployed, and I'll be talking quite a lot about those in vitro diagnostics, but also at the bedside. We also support imaging technologies, from very portable, almost wearable types of technologies that are the next level of complexity up in physics and engineering. I've illustrated here in the lower left a head-only MRI device led by Mike Garwood at the University of Minnesota, but we also support the biggest MRIs with the highest field strengths and the integration of multiple modalities into big physics imaging approaches. We also support therapeutic devices, the delivery of external beams of energy to interior portions of the body. This is an illustration of a focused ultrasound device that can inactivate regions deep inside the brain and cure things like essential tremor. These types of approaches may use ultrasound or RF delivery or light delivery. They can also be energy can be delivered with devices that can be implanted or in a minimally invasive way, for example, with fiber optics. The energy itself can also be used to activate molecules or molecular processes to deliver very efficient activation or inactivation to structures in highly selective areas and in highly specific times. And then this is all tied together with modeling, computation, and machine intelligence. Every one of these examples that I've used to illustrate what we do is either modeled in order to be able to extract more meaningful and valuable information from whatever measurement we make, but also modeled to optimize the design of a technology to do in silico or even twinning, so digital twin types of models that can represent physiologic systems and entire organs and organisms. So that's how we tie everything together. These are all the major technologic foci. And what we try to do is bring all of this into the context of the NIH as a whole. So we're kind of in the middle of all of the institutes and centers. We have 27 of them. It's a little over $40 billion a year enterprise. And what's happened since the formation of NIBIB is that there has become significant adoption of these kinds of approaches and their movement into medicine. So to support that, here's some pretty interesting data. My timeline begins with Francis Collins' arrival in 2008 as the NIH director. NIBIB itself, we have about 1% of the total NIH budget, a little over $400 million a year, but about 13.5, almost 14% of the entire NIH budget is actually spent in bioengineering. So that's been almost a 100% increase since 2008. We compare that with the increase of the NIH budget, a 40% increase. So this rate of increase in expenditures for bioengineering technologies has doubled, more than doubled the rate of increase of the NIH budget itself. And in some areas, like the BRAIN initiative, roughly 2/3 of the expenditures so far have been in the area of bioengineering. Well, the next question, of course, that you would ask if you do like to do modeling and projection is, when is all of the NIH budget going to be bioengineering? And I put this up in jest, but it's also, I think, in partial seriousness as well. In 30 years, we expect that about 90% of the NIH budget would be dedicated to bioengineering. And I think this just underscores what I think all of you know, which is the criticality of technologies in discovery in biology and medicine. And this is something that we continue to rely on. So it's important at the NIH that we have the ability to support non-hypothesis-based technology development, which is where we are with NIBIB. In the pandemic response, as Rick alluded to, we've been able to do this in a pretty spectacular way because of some increased appropriation that came from Congress asking us specifically to address a critical problem of testing. And when roughly around February of 2020, as we started to see SARS-CoV-2 emerge, we started to look at our portfolio and really put together concepts and ideas, proposals to make a contribution to the overall national effort. And our areas of strength where we felt like we could really make a difference were in imaging and artificial intelligence, in vitro diagnostics, and in digital health platforms. So from fiscal year 20 through today, we've seen a really substantial increase, in fact, more than doubling of our base budget, which has essentially forced us to create a second institute. It's almost been like running a lean startup over the past few years, where we've had to hire new people, introduce new processes. It's not easy to spend over $1 billion in such a short period of time. I think probably you know that intuitively. I now know it from direct experience. So let me go through some of these programs that we've created and talk about what the results are and where they're going. The first is in imaging and artificial intelligence. And what we've done is create a program called MIDRIC, Medical Imaging and Data Resource Center. And this allows us to really consider what we can do to help the radiology field in general. Radiologists themselves are really a fundamentally limited resource, and there is an urgent need to introduce AI and ML techniques in radiology for many reasons. One, during the pandemic, with particularly at the time of the launch, we were seeing an overflow in emergency departments and in radiology and imaging suites, where we needed to assess the extent and diagnose disease. Is it COVID? Is it pneumonia? Is it some other infectious process? Monitor therapies, of course, very minimal therapies at the time. We're seeing more of these expand, but this is a critical thing to be able to do in real time and understand phenotypically what's happening. There are complications also that need to be assessed and outcomes that could potentially be predicted from looking at images. And as we've seen over time, post-acute sequelae to COVID or long COVID has become a critical and unfortunately a high frequency event. So it's important to be able to get as much information from imaging and then potentially predict the future and complications for patients. So this center that we were able to form, MIDRIC, is a all hands on deck. Everyone is kind of in, really probably because of COVID, built on our connections and activities that we've nurtured over the years. But with COVID, we were able to bring people together, professional societies, in a way that was absolutely unprecedented. So first of all, on the right, you see RSNA, ACR, AAPM. These are the three major imaging professional societies in our field. They are stakeholders and participants in this entire network. We have 24 academic institutions all around the country. This is led by a group of very talented principal investigators led by Mary Ellen Geiger at the University of Chicago. All of these investigators are at leading academic institutions and affiliated with these professional societies. So because of additional money that we did receive from Congress, we were able to issue a 2-year, $20 million contract for this. It was launched in August 2020. And the primary effort across the network is to build a thoracic imaging and clinical database that's a repository for COVID-19. So the infrastructure that we've built is really an open discovery platform. We have multiple technology development projects that are ongoing. And so far, more than 4 terabytes of data have been ingested. More than 85,000 imaging studies have come into the network. And one of the key things, these imaging studies are like crude oil in some sense, and then they need to be refined to very high-grade gasoline. And so in order to make that happen, they undergo data quality assessment and harmonization, diversity assessment, and we also sequester a portion of these imaging studies for future validation algorithm development. So about 1,000 studies have been sequestered. They are not touched or looked at by anyone until we need to do a validation of a particular algorithm. And then so far, over 20,000 of these have been released to the public. So these are publicly available, curated, very high quality, diverse and representative imaging studies. If you look at midric.org, you can access these yourselves right now. The second major area, in addition to the infrastructure development, has been AI and ML algorithm development. And we have a number of collaborative research projects in this area that multiple institutions are engaging in. We've had more than 24 algorithms developed. More are continued to be under development. They kind of fall into these major categories: segmentation of lung and lung opacities, the prediction of severity and length of hospital stay, and the ability to extract information from radiology reports. And the publication of an algorithmic approach called RADGraph is really a wonderful example of this. One algorithm is currently undergoing a major validation, and this is actually data from Wuhan, but the validation is being done with our sequestered data set that's come from this multi-institutional collaboration. So one of the key things to making data accessible and useful is have some kind of interoperability. So on the left, we have MIDRIC, which is AI-ready medical imaging data. And we view this as an open science type of platform. It's a kind of community metrology, a community effort to develop new tools. But in order to develop new tools, you need to have ground truths. And the ground truths are coming from clinical data, electronic health records, pathology data, genomics data, metabolomics, even mobile health data. We don't have that in the imaging data, but NIH is engaged all across institute-wide, many different efforts to generate this. So some examples are the N3C, the National COVID Cohort Collaborative, that's led by NCATS, the NHLBI Biodata Catalyst, All of Us program, NIGMS's IDEA centers. So they are also creating and building cloud-based repositories of data with a whole set of different data elements that are directly relevant to the imaging needs. So what is our big challenge? It's to bring all of these data sets together, and we're very active in that. We've probably made the most project with N3C and NHLBI's Biodata Catalyst, the PEDAL project. And these are connected in the cloud through APIs, creating workspaces in notebooks and other applications that allow the utilization and further validation of algorithms and discovery from databases that are collected independently, but happen to be at common or overlapping institutions for the exact same patients. Now, figuring out how to connect them and identify what those patients are involves a challenge of developing global identifiers and honest brokers to link the data together so it's all fully anonymized. This is a huge challenge. It's something that all of us at the NIH have been hoping to do. And with COVID and the formation of MIDRIC and these other databases, it looks like we're going to get over the hump and be able to drive some of these things towards formation. Some of the challenges with artificial intelligence have been, and if you look on the right, a couple of very catchy headlines. Hundreds of AI tools have been built to catch COVID. None of them have helped, and that is absolutely true. That came from MIT Technology Review over this past summer. When the world needed it most, AI failed, how COVID-19 poked holes in AI. So these really draw attention to what some of the weaknesses are in some very high profile and major efforts to advance AI and ML. If the data sets are not truly annotated, and I guess using that crude oil analogy, so if they're still pretty much like crude oil and not highly refined gasoline, then there really will be no clean combustion that can take place and utility. So some of these papers on the left point out, and these are coming from the principal investigators in our MIDRIC program, showing that up until now, there are really few public image data sets that have been available. Mostly they're pretty small in size and don't have real world variation. These are some of the weaknesses of the approaches so far. Many of them involve single institution development and single developers. And so the size, diversity, and generalizability of many of the data sets are flawed. Also, we see a lot of concentration of data coming from specific geographic locations, California dominating a lot of the AI and ML data sets and imaging, and as well as New York and Massachusetts. And we've seen this very provocative headline from Aunt Minnie, which is a blog about radiology, sort of posing the question, is radiology AI technology racist? So this work from Judy Gachoya showed that AI can recognize a patient's racial identity on medical images, even though radiologists can't. We're still trying to figure out exactly some of the underlying reasons for this, but I think the bottom line is that it clearly calls out the need for representative data sets. And that's exactly what the MIDRIC program is doing. It's focused, it's really dedicated to high quality, representative, and trustworthy data. And it's building a culture of collaboration that's promoting standards and sharing transparency, best practices. And overall, this is helping us accelerate the translation of AI and ML into real world approaches where we can potentially develop algorithms to quantify how medical imaging is working. And we have an opportunity here to build on this general type of structure and extend to other imaging modalities, organs, and diseases. So that's what we're doing in medical imaging. And now I'd like to shift to in vitro diagnostics. I guess the other extreme end of the scale in terms of cost and accessibility. So medical imaging is, generally thought to be a big physics type of platform that you bring patients to. And in vitro diagnostics are small, portable, essentially very low cost platforms that can be brought directly to patients. We can have them in the home now with over-the-counter tests and their emergence. A key to making a lot of this stuff happen are digital health platforms, and I'll go into a little bit of what we've done, some of the challenges are in digital health platforms. This whole program emerged, it was essentially an unexpected opportunity for us. It emerged in April of 2020 with the 4th congressional supplement that appropriated $1.5 billion to the NIH and led to the appearance, the formation for the first time of the RADx family of programs, RADx Tech, RADx ATP, RADx Rad, which Rick is spearheading, and also then works with colleagues, and he and I both are working together on the executive committee for all of these RADx programs, including RADx UP, which is dedicated to underserved, moving diagnostic technologies into underserved populations. There are more than a dozen NIH institutes, centers, and offices widely engaged in all of these programs. From the RADx Tech and ATP point of view, the technology development part, what our mission has been is to expand testing technologies, their number, type, accessibility, and really optimize their performance. So I've been co-leading this with Jill Heemskirk, the Deputy Director of the NIBIB, and immediately we formed a partnership with BARDA, with the Assistant Secretary of Health, the FDA, Department of Defense, the White House, the CDC, recognizing that the ambition here was to really transform how diagnostic technologies were done. And that wasn't just about the technologies themselves. It required quite a heavy lift in the regulatory side and the implementation side and the coordination of multiple government agencies to make this happen. Hence, the leadership from the Office of the Director, so Francis Collins himself, Larry Tabak, and Tara Schwetz, Rachel Florence, have formed the core team to guide all of these diagnostic technologies with a regular cadence of meetings that continue to go on, even though Francis has now stepped down as director. We were able to get this program off the ground. The appropriation, if you're paying attention to the dates, was on April 24th, 2020, and we managed to get the program going on April 29th. So 5 days after the launch, after the appropriation, we were able to launch a program. The only reason why we could do that is because we built on an existing network, what we call the Point of Care Technologies Network. It's a U54 that we've supported for several years, but we expanded the network dramatically. So nearly 1,000 experts, we sort of sent out a call to the innovation and entrepreneurial community and said, we need help. And we got lots of response. We were able to hire several expert consultants by building into the network 2 not-for-profits, VentureWell and Biocom, 2 organizations that we've worked with extensively before. So we could fund the nonprofits and then they could hire the expert contributors from industry to help provide key operational roles that we were going to have to accomplish. And I'll go into a few more details on exactly what's done through the network. But fundamentally, this is composed of 5 major universities, Georgia Tech and Emory, CIMMYT MGH, which is the coordinating center at the core of this, Johns Hopkins, UMass, and Northwestern. And within these 5 academic structures, we also were able to carve out and identify very unique and specific roles: a validation core, which is at Emory, a clinical studies core, which is at UMass, and a deployment core, which is housed at CIMMYT. So overall, with all of these experts and the universities and the formation of multiple committees and workflows, the operations have been to both review and fund ideas that would come into the network. So where are we going to get the new technologies? We solicited innovators and entrepreneurs to send us ideas, and then we reviewed and funded them. But every idea that was funded had to be tested and validated, and that was done in the validation core and in the clinical studies core. And throughout the entire process, it wasn't a typical process where you submit a grant proposal and then we check on you several months later. We actually formed teams around every good idea, and those teams provided expert guidance in everything from how the technology would work to how you can commercialize, find markets, and get to manufacturing. And those daily cadences, I think, were the difference between success and failure of many of the technologies. So the overarching structure, the process for making all this happen is what we call an innovation funnel. We had about 3,000 applications started. We ended up having 824 full applications come into the innovation funnel. And these came from a diversity of sources, small businesses being the dominant one, over 400 of those, but also academic groups, startups, mid-sized businesses, even large businesses. Over 50 of these applied to the RADx network. We had two distinct funnel periods where we opened up 4 applications. The first was in May of 2020, that lasted about 3.5 months. The second in June, that was 1 month. And we had a rolling submission process during those windows. The goal was, from the time we opened the funnel, to be able to have a series of reviews. And many of you may have heard of the Shark Tank. So that was our first phase, the first phase of review, where essentially every technology that made it into the Shark Tank, every group, went through roughly a week to 2-week analysis, detailed analysis of the strengths and the weaknesses of that approach. If they made it past that, they were advanced to a first phase of funding that lasted on the order of about 2 months, maybe about $1 million. And if they made it through that, those groups would advance to a second phase of funding where they were expected to go all the way through regulatory, manufacturing, and distribution. And we've had about 50 make it through to there. We've spent about $700 million on those 50. And if you just sort of take, here's a snapshot and an almost complete list, but you can see on our website where we describe all of the phase 2 awards. There have been, we sort of broadly separate these into point of care and home technologies, laboratory-based technologies, both new technology itself. For example, there's a platform called Quanterix, which is a single molecule-based antigen detection technology that uses femtowells and an imaging camera. It does not require amplification. It actually has a limit of detection that's comparable to nucleic acid. It's very high throughput, but it's all antigen-based. Fluidigm, I think many of you are familiar with that laboratory technology. It's an integrated fluidics chip. From a point of care and home point of view, those include both antigen assays like Quidel QuickView or the Illum assay, but they also include point of care RT-PCR like that came from Mesa Biotech, which is a small startup in San Diego that eventually was bought out by Thermo Fisher for about $500 million, or Visby Medical, which is another small handheld RT-PCR device. We also have supported a network of laboratories that are super labs in a sense. So the Broad Institute, which was able to do about 100,000 tests a day at its peak period. And we supported innovations in automation and workflow advancements to be able to get to that point. Labs like Helix. So both of these well-established laboratories that we were able to expand their capabilities so we could get to the point where very high throughput, high capacity could be done in certain sites all around the country. And we've also supported laboratory products. For example, Mammoth Biosciences CRISPR approach, Ceres Nanosciences, they make these nanoparticle pre-concentrating they're nanoparticles that can concentrate a sample by through the structure of the nanoparticle itself and improve limits of detection anywhere between 2 and 10-fold for a particular assay. Also swabs, injection molded approaches, as well as saliva collection devices. So we kind of covered the support of virtually all the products in this field. And well, let's see, maybe I missed a slide. Okay, it's coming. We've also formed a new process with the FDA to try to accelerate authorizations. That's called ITAP. That's the Independent Test Assessment Program, where again, companies will come in, we evaluate them using an intensive review process and determine what they need in order to get their EUAs together. And then we've had special additional funding for manufacturers that can provide very, very high volume. For example, we've supported Siemens and Roche, and their tests came into this process. This is more about process innovation as opposed to technologic innovation. But in less than 2 months, we were able to bring both of those projects to authorization, adding more than 100 million tests to the monthly test capacity, which is something that we urgently needed over the past few months. And we have several of these that are still in process. So this has been a very, very important contributor to the increasing the number of tests and test capacity. So if I add all of this up and we go through December of 2021, we've increased the testing capacity for the country by 1.8 billion tests. That's about 8.3 million tests and test products per day in December, with 39 EUAs, emergency use authorizations. 7 of these have been for over-the-counter tests. 6 of them have been for antigen tests, but 1 of them has been for an over-the-counter PCR and RT LAMP process from a company called Detect. So in December, we were doing about 250 million tests. If you go back to 5 months after launch, what we had hoped was to be able to move the needle on increasing test capacity, and we had increased it by 20 million in September of 2020. And if you go back to April of 2020 when we launched, the country as a whole was only doing about 5 million tests, 6 million tests in the month, and they were all laboratory tests. So we really hit our goal, which was to expand all forms of tests and test products and dramatically increase the cumulative test capacity. Of course, we had no idea that we would be in this for so long and that we would need to increase the cumulative test capacity by so much. If we just take this snapshot of December 2021, the contributions of NIH to increasing test capacity, this has been a little over half of the total test capacity for the country. Well, increasing the capacity is one part, but implementing it and figuring out how to provide guidance on when people should test is another major part. And we've worked with our POCTERN network, and in particular with CIMMYT and MIT to develop a calculator that can guide individuals. So there's a stepwise decision assist device that's available for individuals on whentotest.org. It's an individual risk calculator. You can also do this exact same thing for organizations. It takes into consideration your vaccination status, masking, your location, the prevalence there, whether or not you plan a gathering or have had one recently in the past. We're able to update this on a continuous basis for R0s for specific variants. And there are very well-organized playbooks, for example, a K-12 playbook that the CDC also shares with schools around the country. And this helps guide organizations for how they may set up testing programs. There's even a link on this for purchasing and further guidance on the use of all the different technologies. So I encourage you to take a look at whentotest.org. We've also, at the beginning, when we were starting to create and support over-the-counter tests and really push for home use, no one had done home tests at any kind of scale before. So we launched a pilot program, a website that really has been the precursor for the current USG website for the distribution of tests called Say Yes COVID Test. This has been a partnership between NIH and CDC, as well as Duke and UNC, their centers that are part of the RADx UP, the coordinating centers that are part of the RADx UP program, and have rolled out online distribution, so entirely online, driven by a digital health platform, of about 7 million over-the-counter tests in 10 states, nearly 800 cities all around the country. And at the core of it is this platform developed by Care Evolution that allows full online fulfillment and linkage to Amazon in order to be able to get your test delivered directly to your door. At the back end of this, we have opportunities for people to do reporting. And one of the weaknesses in reporting of home tests is that there have been no data standards at all and no data hubs to collect all that information. So we had to launch all of those and form partnerships with the APHL, the American Public Health Laboratories, the Office of the National Coordinator, and the FDA. And now we have the highways that are built with the data standards, and we're able to potentially move information from home tests to hubs and networks, go to state and federal public health authorities. And what's really exciting about this is now that we are on the brink of being able to deliver therapies, antiviral therapies, for example, to people in their homes, this type of platform can serve as the foundation for test-to-treat paradigms that we're really hoping to be able to deploy in COVID and, of course, in other diseases. Everyone is really concerned about variants, and ultimately this is what is the key problem and the persistence of the pandemic. And we started a task force on variants early in 2021. And the goal of this was to use a bioinformatics approach to be able to follow in GISAID all of the mutations and how they are they can be found in variants of different lineages. And then as each one appeared, to be able to do a computational analysis of our PCR tests and assess which ones are likely to fail or be impaired with the appearance of these mutations. So in addition to looking at variations in performance in nucleic acid tests, we're also able to extrapolate to performance in antigen tests. And if it looks like there is likely to be an impact, then we go through a rigorous process of evaluating the performance of these tests in the laboratory with actual variant samples. And those labs that are doing that are primarily at the validation core at Emory and Georgia Tech. So this is, again, a broad federal agency collaboration. It involves first informatics-based screening and then banking samples. So there are more than 125,000 samples now at Emory that are banked and available for analysis and wet lab testing. And we've systematically moved through every FDA authorized test to be sure that there is no significant impact in performance. The flip side of this task force is if we're looking at how variants can potentially impact performance, it may be possible to design tests that specifically detect variants more efficiently or detect any SARS-CoV-2 without regard to its variant status. And that's led to what we call Project ROSA, which is the development of a SNP chip or a genotyping approach that can identify variants. And in this version of the SNP chip, which is a 4-marker reflex genotyping test, it can distinguish between Omicron and Delta. So really within weeks of the appearance of Omicron, we had this dashboard up, and working with Thermo Fisher, had the primers and the plates ready to send out to labs around the country. There are a few labs that are running it that are RADx supported, but now some other labs are signing on as they've looked at exactly how this works. So there's a meta-archive publication that refers to this. And Rick, I think you might want to send that around if people are interested in seeing what that linkage is. It provides all of the information if you'd like to reproduce this in your own laboratory or know if people would like to do that. This also includes BA2. So within about a week of identifying BA2, we were able to stand up and add an additional marker in this reflex genotyping approach so that it can distinguish between BA2/3 and BA1. And we're tracking this on the dashboard. So again, you can go to this URL and track it. It's updated a couple times a week. One of the unique features of the SNP chip as compared to NGS, which is our alternative approach for looking at variants, well, it is the foundational approach, is the speed and the cost. So the turnaround time on this test is really a few hours. And the cost of the instrumentation, any lab that's running a Thermo platform can immediately run this, but any lab that's doing any type of genotyping could potentially create an assay themselves. It's accessible, therefore, to most labs all around the country, all around the world. We can modify it with new variant integration in less than 2 weeks and then validate it. So this has been a fantastic partnership that we've had with Rosalind, the bioinformatics company, Thermo, these laboratory sites, Helix, Aegis, University of Washington, as well as Emory and GISAID. So where are we going? What are some of the future directions and challenges? I think if you're following all of the questions and debate about lateral flow assays or rapid antigen tests versus PCR, you recognize that there is this enormous gap in terms of their limits of detection. And we see that antigen tests have limits of detection that are on the order of about 1 million copies per mil. Laboratory tests or even point of care PCR tests have limits of detection that are about 1,000 copies per mil. So that 3 orders of magnitude gap is what's creating a lot of confusion about the use and the guidance of rapid antigen tests, how sensitive they are, how frequently you should use them. Are they really any good at all? And we are continuing to develop technology to bridge that gap. A lot of the really interesting innovations are in nanoscience. We've seen well over 100 applications coming into the RADx innovation funnel that are predicated, that are based on nanomaterials, from nanopores and quantum dots to nanotubes, nanowires, DNA nanostructures. So this has been very exciting to see. We funded quite a few nanoscience approaches. About 18% of our phase 2 funding has actually been in nanoscience. And some of the more notable examples, the Illum test uses an integrated optoelectronics device with a Bluetooth chip. It has light source, detector, and optics all inside of a small plastic cartridge. And the readout of the immunoassay is accomplished by exciting fluorescence in quantum dots. So quantum dots are on the shelves of CVS and many local drugstores all around, something I would have never anticipated. This is one of many. We've had hundreds of millions of nanoscience-based technologies now sold. Another really huge performer is, as I mentioned before, the Series Nanoscience pre-concentrating the sample concentrating device that's widely used in a number of assays. But there are a number of these that are still in progress that are being developed. One that we've recently funded is essentially a 3D nanoelectronic biosensor. It's built on 3D flash memory or VNAND types of structures, silicon structures that have nanopores. So potentially hundreds of thousands or millions of these addressable nanopores that inside of the nanopore, what you've got is a DNA probe that's immobilized and there's no amplification. So the binding itself creates current. It's essentially like a field effect transistor, and they're addressable. And in the future, it may be that some of these very portable home-based devices will look very much like consumer electronic devices. So all of this stuff has really made an impact, and I think all of you have been following the Biden administration's announcement of first 500 million tests and then 500 million more, so 1 billion tests to be distributed, something that we have been pushing for within the RADx program for many months. And these tests are being distributed now through a national website, and many of our programs, the Innovation Funnel and the Say Yes COVID test with our digital health platform, our ability to authorize these rapidly at scale and at volume have been the prototype programs to make a lot of this stuff work. So what are some ongoing challenges? We wanted to drive a paradigm shift, and be careful what you wish for. So we started with laboratory technologies far in excess of point of care technologies. As I mentioned before, lab technologies were accounting for about 5 million tests in April. And now we see in December, there were about 460 million tests done. 300 million of these were OTC, over-the-counter tests. About 50% of these tests have been RADx-supported tests. And point of care and over-the-counter far exceeds laboratory. Laboratory is an increasingly small fraction, roughly 15%, but getting smaller and smaller as we progress. That means the guidance, I mean, it's kind of easy if you're in a laboratory, a physician prescribes it, is working with you, interprets it, but you can't really access that many people, and it's not that good for screening and surveillance. So now we have tests in people's homes. We have hundreds of millions of users, and they're all talking on Facebook and social media about the tests and how do they work. And so it really cranks up, I guess, the pressure on us to be able to communicate the performance of the tests and explain it in simple terms. So this plot here on the right is anything but simple terms, but I'll just try to walk you through it. This is an ROC curve, a receiver operator curve for antigen test performance. It's probability of performance on the y-axis versus CT count on the x-axis for a particular Roche PCR device. And what you can see, and this is in over 500 subjects with Omicron, so this is actually done only with Omicron patients, you can see that the probability of antigen test performance of getting a correct answer is decreasing as you go to higher and higher CT values as the viral loads go down. Not surprising, but it just says essentially antigen tests work really well when there's a high viral load. But if the viral load is low, like it may be in the early days of an infection, and the CT counts are very high, you have weak positives, then antigen tests are not that good. So if you look on this curve, you see the serial test arrow pointing up. This means we want to get up this curve. So the idea is take another test, do it again 24, 36, 48 hours later. And what we find is typically after the onset of infection within 48 hours, you move up into this region of high sensitivity and high viral load. So that's one of the tricks with antigen tests. They're inexpensive and accessible, or they need to be, and they should be used more frequently. So if you get a negative on an antigen test, you need to presume negative, but test again. And that's a key part of the guidance that has been difficult to communicate to the public, because everybody wants to take a test, get a negative, and say they're clear, and that's done. But that's not necessarily the case. We also see that the cost of antigen tests are a continuing challenge. In December, and to some extent in January, certainly November, December, and part of January, the demand has far exceeded the supply. So the cost of the tests have been really too high to afford for most people this serial testing concept. So in principle, you want to have excess tests where the supply far exceeds the demand and people can use them quite liberally. So this reverse is going to be happening just really as we speak. We are projecting about 1 billion over-the-counter rapid antigen tests to be available in March. And this is an incredible scaling up from the 480 million, a little over half of that, tests that have been available in January. The part of the trick here, though, is to how do you sustain that? Because the test manufacturing industry responds to the demand. So if there is a drop in demand, as we're seeing when the case reports drop, then we don't have as many tests because the manufacturers pull back and they don't make as many. So one of the challenges is to figure out how to invest in innovation where we can sustain this and create really true low price points, $2 to $4 in retail price, so that tests can be easily accessible all the time. We can keep a kind of warm base going for other pathogens and diseases and be able to crank it up quickly within the United States to be able to deploy them fast. Other challenges are improving performance information content. As I mentioned, there's this large gap between the limit of detection of antigen tests and laboratory tests. What if we could have antigen-like form factors and cost, but with laboratory performance? And what if we could do it for more than just COVID, other respiratory diseases, other viruses, and other conditions? And we also want to have more quantitative measures of response to vaccines and therapies and long COVID as well. So there's more things to measure, more things to test in terms of the information content, and we need better performance. All of this is moving into the paradigm of test treat, where information can move from home to physicians with telemedicine and really optimize therapy for individual patients. And finally, better screening and surveillance. We've shown with our experiment in Project ROSA that you can go with very fast turnaround time with the identification of new variants and pathogens, be able to stand up new tests very quickly. But this is really an NIH demonstration project, and it's not something that's been widely embraced by public health. So hopefully this can be adopted. Wastewater, air sampling, breath analysis, ways to trigger test to treat paradigms. These are all very active, cutting edge areas in the testing universe. So to try to summarize all of this, we've done a rapid conversion of ideas and technology to disseminated products and solutions. I think something that we're not too used to within the NIH. We're generally thinking, all of us in academics and in our world, of 5 to 10 year cycles to be able to do this, but we really had no choice. I mean, we really needed to pressure this and make it happen in a very short period of time. So we've been able to advance both technologies as well as processes to do that. The process innovation probably in some ways has been the more surprising and necessary element of innovation because there's a lot of resistance to these kinds of changes. Perhaps without the driving pressure of having a global pandemic, we may not have been able to launch this kind of process innovation so quickly. For the future, we're hoping, and I think all of government is really looking at, how will we sustain all of the work that's been done, not just RADx, but all of the work that's been done in vaccines, in therapies, in diagnostics and testing, so that we can be ready for the next pandemic and also take advantage of many of these structures so that we can move it into slower moving diseases, cancer, cardiovascular disease, metabolic disease, and so forth. So finally, I'll just sort of leave you with this excerpt from a Nature Biotechnology editorial that in April pointed out that RADx has radically shifted funding available for innovative diagnostics. And I think this is broadly in vitro, but it also includes medical imaging and foreshortened product development times. But this is kind of the challenge that the editor has laid down for us. For too long, we've talked the talk of precision medicine. Now's the time to walk the walk. So I think we've shown that elements of that can happen. Really, the next question is about the sustainability of all of it. So I'll stop the talk there, and I think we may have some time for questions.
We're running a little bit late, but well, we actually have a few minutes here. But Bruce, I mean, that was absolutely fabulous. I just kind of watching some of the private chats coming into me. People are really impressed. But I'm hoping that, and Lynn will get to you in a second here, I'm just hoping that we can bring some of the remarkable capabilities that you have at NIBIB to work with us on developing rapid tests and public-private partnerships and other things around environmental testing. So I think you can bring a lot of innovation to that whole area. Lynn, you have a question or a comment? Lynn Goldman from. Go ahead, Lynn.
Hi, thank you, Rick. Yeah, it's a little bit of a comment and a question for you. So we in public health definitely, appreciate the role of the testing in clinical management of COVID and of course other things, but also there's a population health issue that it's extremely frustrating, and that is that we have no idea from the data that we have about what the true prevalence of this thing is across our communities. And in some ways, to be honest, I'd rather test people less frequently and have more reliable results that are reportable, so I know what's going on from the standpoint, of being a public health official, from the standpoint of being responsible for population health, to be able to understand, truly what the variability is and what the trends are. And so, I mean, we have data across our communities for test positivity, which, as you know, is completely nonsensical because the availability of testing is so variable, and the decision to whether to go get a test. And now with the home testing, it's become even worse because people don't report the results to public health. So I just wanted to make that as a comment, but I was also wondering if that's a consideration at all in this. I mean, I certainly didn't see it in the model that you presented, which is really based, I mean, we use models like that on our campus and stuff, those of us who are involved locally, in our own communities, but I didn't see that population health consideration in that.
Well, it's a great point and one that is intensely debated. So from a technology point of view, all we can do are create the highways for reporting. So we have created those now. We've worked with the ONC to identify and agree on common data elements for home test reporting. We've worked with APHL to create the connections to states, and we have the data hubs. We have there has been no structured data language that comes from those tests prior to this experience. So we can now transmit and fire in HL7, V2, although not all states can accept HL7. Most of them are accepting faxes and phone calls. So, I think that there's a technologic component to it, and then there is a structural component where I would love to see the public health community gather around that. We have, from the experiments that we've done in small scale, found that the people who do decide to report, which has been, again, a very small pilot study that we've done in Say Yes COVID Test, they report with vigor, and we can get a lot of information from those folks. There are also options to report completely anonymously. So we have a lot of different options for reporting. And whether we like it or not, the home tests are the dominant platform, and we'll be looking at 1 billion home tests that are distributed. We would love to be able to get reports from those, and the discussions are occurring at many different levels on how to activate the switch to create visible reporting pathways. Some states have launched those themselves, not many, but from a technologic point of view, to get information standardized, manufacturers are they're all very eager to participate with us, and to create the highways for that. That's all happening. And I think the long-term benefits, it may take a little bit to get it jump-started, but the long-term benefits for both public health as well as personal health will be enormous.
So I see that Katrina made a comment in the chat box about the wastewater testing. And so I'll just encourage everyone to take a peek at that. Katrina, do you want to make a quick comment?
I was just thinking about how after class yesterday, one of my students came up to me and said, we're reporting 1 positive student, but I personally know 6 who are sick and won't report their results because the consequences are so draconian. Multiply that at the community scale, and it just makes sense that the colleges are trusting their wastewater results much more than their reported student clinical tests.
Yeah, and I look to you as experts in public health to really figure out the right structures and incentives. I think this is a classic opportunity for collaboration with the technology community. I think we can create the highways, the structures, and think through how to get the data out of, the home test to wherever you want it to go. But this is where the partnership has to happen to think about how to encourage and incentivize all that.
Sounds good. Pat, do we have time to slip in one more question from Phil Bourne?
Yes, let's do it.
Great. Phil, you're on.
Yeah, thanks. And thanks, Bruce. That was great. I have a sort of question that sort of steps back from the specifics of the 3 areas you talked about, and it relates to training. So it strikes me that some of the things that you talked about, the use of ML and imaging, for example, and then the health platforms and everything that was described around the in vitro diagnostics, there seems to be a growing gap between what some of the things you described and how we're training people to do these things. I worry about that in the context of now being in an academic medical center. I just feel our curricula and our programs, and there are pockets where, for example, particularly in bioengineering departments, things are going pretty well. But if you look more broadly across our training, I worry that there's a widening gap between what can be done and what we're training people to do.
Absolutely. And that's why I think that there's a lot of enthusiasm for bioengineering departments within departments to reach out and form partnerships in a lot of ways. And this touches on the public health conversation. This is the first time we've seen a wider engagement of engineering and technology developers with public health at this kind of scale. And I think it's important for bioengineers, technology developers to at least have a seat at the table. We've been fortunate, just for whatever reason, Congress kind of called on us to do this, there was a recognition that there was a national crisis pending in the availability of tests and that we needed innovation and entrepreneurship to drive that. So we've seen that's uncovered a bit of a weakness in how tests have been developed and reported and deployed in the past. All of these new partnerships, we hope we can continue to build on them. And I hope all of you can hire bioengineers. So we have a lot of fantastic students. A big part of what I do when I go around and speak to biomedical engineering departments is speak to the importance of their formation of partnerships with public health. I think this is the new horizon for us. There are many, many opportunities to make a powerful contribution.
Bruce, that was just absolutely terrific. So I think just looking at the clock, Pat, we're going to have to move on. Is that correct?
Sure.
Okay. So Bruce, thank you. Really appreciate it. And so we'll be in touch and keep up the great work on RadX Tech and a whole variety of other things that are happening at NIBIB.
Thank you so much. It's all great to talk to all of you.
Thanks for joining us.
Rick, we have next up, Daryl's going to give us a little background on, going to provide an introduction to Dr. Shaw and give us a little background on a program that we're involved with.
I think, Daryl, I think you're on. So you want to share your...
Yeah, can
Good.
you see me okay? And can you hear me okay?
We can see and hear you. Great.
Good.
Go.
So I'm going to give a brief summary of the Lasker Clinical Research Scholars Program and then introduce Natalie. So for those of you who don't know this program, it's a program that focuses on career development for physician scientists. So the goals are to develop leaders in clinical research by supporting selected researchers early in their independent careers, and to allow selected clinical researchers to take advantage of the intramural research program environment and resources, but at the same time establishing themselves as peer-reviewed NIH-funded investigators. So it's sort of a hybrid program. So the candidates for this program are early stage clinical researchers, so typically physician scientists or nurse scientists that are not yet tenured, with the ability to conduct independent research. And again, physicians, dentists, or nurse researchers are eligible. The program is an 8 to 10 year program divided into 2 phases. So in the first phase, the NIH Intramural Program provides full support for the investigator, including salary, staff in their laboratories, and research support. And they're considered an independent tenure track investigator. And typically this phase lasts between 5 and 7 years. And then phase 2 of the program is an R00 phase, where the salary and research support of up to $500,000 a year are given by the Institute extramurally to an outside organization. And they can get up to 5 years of that money. So the funding, the funds for the first part of the program are intramural funds, and for the second part of the program are extramural funds. And so and they're all done through a particular institute or center. There's average of 4 to 6 scholars per year across all of NIH. So there are a number of benefits of the program to human health, to extramural NIH, and to intramural NIH. To human health, promising clinical researchers are encouraged to pursue a career in biomedical research. To the extramural world, the intramural program provides the bulk of funding early in the career of these individuals. And to the intramural NIH, it's an attractive environment because these scholars perceive that they have many more career options. So they can elect to stay intramural after the first part of the program, or they can elect to go extramural. The benefits to the scholars include an outstanding intramural environment for clinical research. They get to take advantage of the NIH Clinical Center, the nation's largest research hospital. They support transition from laboratory to clinic, and you'll see this firsthand in Natalie Shaw's presentation. They get access to state-of-the-art resources, including imaging, molecular analyses, and other laboratory facilities, and a favorable regulatory environment with centralized IRB. They also get protected time for the intramural part of the research, so they don't have to write grants, they don't have to teach, and they are protected from providing patient care during that time. So it's fully funded by the NIH Intramural Program. And then the extramural component provides another option for scholars, if they so choose, to get support at a research institution of their choosing after they complete the intramural phase. So status of the program. So the program is just about 10 years old. To date, there have been about 38 Lasker scholars that are either enrolled in the program now or have been in the program. 5 of them so far have already been tenured, and their names are listed here. One of them was just elected into the ASCI, American Society for Clinical Investigation, and several are now in tenured positions at outside organizations. And only one scholar left the program, but left for a leadership position at a large biopharmaceutical firm. This is the breakdown of the Lasker scholars by institute. Most institutes have 1 to 3 Lasker scholars. We have 1. A few of the larger institutes have very actively used the Lasker program. And as you can see by the pictures of the current Lasker scholars, it's a very diverse group. More than half are women, and a significant percentage are underrepresented scientists. Here's Natalie Shaw, who's our Lasker scholar. So maybe before I introduce her, I will stop sharing my screen and, if necessary, answer any questions that you might have about the Lasker Scholar Program. Are there any questions? Oh. If not,
it looks like they're interested in hearing Natalie's work.
All right, let me introduce Natalie then. So Natalie is our Lasker research scholar. She's the head of the pediatric neuroendocrinology group in our clinical research branch. Dr. Shaw earned her medical degree from University of Buffalo School of Medicine, and then a master's in medical science from Harvard Medical School. She completed her pediatrics residency at Children's Hospital of Pittsburgh, and then a pediatric endocrinology fellowship at Boston's Children's Hospital, and then a clinical research fellowship in the reproductive endocrine unit at Mass General Hospital in Boston. She was an attending physician at Boston Children's Hospital before we recruited her here to NIH in 2015. So as a pediatric endocrinologist, she studies environmental and genetic control of pubertal development. She conducts studies in healthy pediatric volunteers that are complemented by genotypic and deep phenotypic studies in patients with rare forms of hypogonadism, as well as studies in patient-derived neural stem cells that you'll hear about today. And her talk today is entitled SMCHD1 and Epigenetics, Lessons Learned from Patients Born Without a Nose. I'll turn things over to Natalie.
Okay, great. Thank you so much for having me here today. Let's make sure I have control. So as with many medical discoveries, this one started with an intriguing patient. So this patient was a 23-year-old female who presented to the reproductive endocrine unit at Mass General Hospital, where I was a research fellow, with primary amenorrhea or absent menstrual periods. She reported that she had been born with multiple midline defects, including the complete absence of an external nose, a condition called Irania, and some type of hypothalamic pituitary defect. So what do these two conditions have to do with each other? Well, before I answer that, let me just briefly back up and review the components of the reproductive axis. So the axis is controlled by a network of gonadotropin releasing hormone, or GnRH neurons, at the base of the hypothalamus. GnRH stimulates secretion of LH and FSH by the pituitary gland, which then stimulates sex steroid secretion by the gonads. Now, because reproduction is so energy consuming, particularly for women, it's tightly controlled by a number of internal and external factors. For example, metabolic cues must indicate sufficient energy stores, and environmental cues must indicate favorable conditions for reproduction. There's also a strong genetic component to the timing of puberty and its ongoing function. Now, the axis has two very unique features. The first is that GnRH must be released in a pulsatile fashion, as shown here. I'm not sure if you can see my mouse. And we can't measure GnRH in the blood because of its very short half-life, but we can measure LH, which is a validated marker of GnRH secretion and is also pulsatile. The second unique feature is that GnRH neurons don't actually start out life in the brain, they start out life in the nose. This is a close-up of the roof of the nasal cavity, showing that it's separated from the brain by, and in particular the olfactory bulb, by a structure called the cribriform plate. Now, very early in development, an ectodermal thickening called the olfactory placode gives rise to olfactory neurons, which extend their axons through the cribriform plate into the brain, synapsing on the olfactory bulb. The placode also gives rise to GnRH neurons, which hitch a ride along these axons to eventually reach their rightful place in the hypothalamus. Now, as this is all happening, however, the cribriform plate is more of like a soupy mesenchyme. And so when it solidifies into bone, as shown here, this is the base of a human skull, you can see these holes in the cribriform plate that represent olfactory nerve passage. And so, as you might expect, if there's a defect in olfactory structures, there is also going to be a defect in GnRH neuronal ontogeny. And there's a name for that combined defect called Kaufman syndrome. And these patients have olfactory defects, so they're born without olfactory bulbs or tracts, and they have congenital anosmia, or an absent sense of smell. And this is something that can be tested with a simple scratch and sniff smell test shown here. They also have GnRH deficiency, which manifests as micropenis and cryptorchidism, meaning undescended testicles in males, and absent puberty and infertility in both sexes. Now, if we were to do a frequent blood sampling study in a patient with Kaufman syndrome, shown here, we would see extremely low levels of LH, in contrast to this more robust pulsatile pattern of LH shown for a normal male. And studies in patients with Kaufman syndrome have uncovered mutations in more than 30 genes that play critical roles in controlling reproductive function. And many of these same genes have shown up in genome-wide association studies for ages at menarche and menopause. So does our patient simply have an extreme form of Kaufman syndrome? Well, let me tell you a little bit more about her presentation. So at birth, she had an absent nose, she had microathalmia, or very small eyes, as well as cataracts and coloboma, which is a keyhole defect in the iris. At age 16, she had no signs of pubertal development, so no breast development, no menses, and she had very small ovaries and a uterus on ultrasound. She underwent a clinical evaluation of her reproductive axis, which demonstrated extremely low levels of LH and FSH, but the remainder of her hypothalamic pituitary axis was intact. At age 23, she also had a formal smell test, which led to the diagnosis of a complete anosmia, and a brain MRI that showed complete absence of the olfactory bulbs. She was also one of several patients who we flew to the NIH Clinical Center to have testing at NIDCR called a cone beam CT, which lets us take a very close look at some of the skeletal structures of the face and jaw. And shown here is a cross section through the base of the skull, and you can see the aerated ethmoid sinuses and tiny orange arrows, which are pointing to holes in the cribriform plate. This is in a healthy volunteer. And here's our patient, in which this entire region is like sheetrock. So again, no evidence of olfactory or GnRH nerve passage. We performed genetic studies in this patient, but this was negative for all of the known Kallman genes. So what does this patient have? Well, when I encountered her as a fellow and did a bit of digging in the literature, I realized that she actually had a little known syndrome called Bosma Irania Microathalmia syndrome. This was first delineated by a Dr. James Bosma at what was called the National Institute of Dental Research in the 1980s. And he came across two male cases with the very same striking triad of clinical features: Irania, ocular defects, and hypogonadism. Irania is exceedingly rare. And at the time I first saw this patient, there were fewer than 100 reported cases in the literature over the past century. But the presence of familial cases and the absence of any unifying environmental exposure suggested that there could be a key genetic component yet to be discovered. So partnering with this patient, who was very curious and very bright, very curious about learning the etiology of her condition, I set out to try and determine the genetic cause. And it turned out by pure chance, her mother had kept a newspaper clipping of a local man with the same syndrome. And I was able to find him through some Google detective work. And this is a picture of him in his 70s. He's since passed away, but he was working as a lawyer in Albany. And it turns out he was actually one of the first patients described by Dr. James Bosma in his 1981 report. So then, partnering with other endocrinologists and clinical geneticists and contacting authors of previous case reports, I was able to start to assemble a cohort. And I was even able to enlist the help of a mother shown here who runs an Irania Facebook group. So through that effort, I built a cohort of 45 cases and 51 family members. Next, partnering with colleagues at Mass General, we performed whole exome or whole genome sequencing on the first 21 patients and looked for ultra-rare non-synonymous variants. We found that 84% of cases had a mutation in the same gene, the gene SMCHD1, and these were all heterozygous missense variants restricted to exons 3 through 13 of this very large 48 exon gene. And just to confirm this statistically, we also performed gene-based rare variant burden testing, where we looked at the frequency of rare variants in every gene in the genome in our cohort compared to 60,000 controls in what at the time was called the exact database. It's now referred to as NOMAD. But as you can see in this Manhattan plot, there was just one gene to achieve genome-wide significance, chromosome 18, and that was again SMCHD1. But we had a slight problem. And that problem was that mutations in this same gene, in some cases identical mutations, had already been known to cause a very different disorder called facioscapulohumeral muscular dystrophy type 2. Now, this is a degenerative muscle disease with average onset in the 20s, and it causes slowly progressive weakness of the face, shoulder girdle, and upper arms. This is a picture of a man with something called winged scapula, where the muscles that normally keep the shoulder blade anchored to the rib cage are weakened, causing this wing-like protuberance. Now, it's not unheard of for mutations in one part of a protein to cause one disorder and mutations in another part of the protein to cause a different disorder. But as I'll show you here, this is not the case in Irania and FSHD2. There's instead significant mutational overlap. So this is showing a schematic of the protein, and here the 48 exons at the bottom, and the different mutation types. So red are deletions, gray nonsense, splice sites, and then black are missense variants. And the SMCHD1 protein has a ubiquitin-like domain at the N-terminal end, an extended ATPase domain, and a hinge domain. And you can see that in FSHD2, many of the mutations are loss of function and they span the entire protein, whereas the missense mutations tend to be clustered in the extended ATPase domain. And the same is true for Irania. Here, all of the mutations are heterozygous, missense, and restricted to the extended ATPase domain. If we look at the tertiary structure, we can see that same level of clustering. So in collaboration with Lars Peterson and the NIH crystallography core, we were able to crystallize an N-terminal fragment of the protein from amino acids 24 to 580. You can see that the construct dimerizes. So there's a swapping of this strap shown in purple and the ubiquitin-like domain shown in beige. And if you look at the mutations, the magenta are the Irania mutations, blue are FSHD2, and the orange are the mutations seen in both conditions. But the mutations in both disorders tend to lie along within the ATPase domain and at the dimer interface. So you may be asking yourself then, is this one disease or two? That is, do patients with Irania go on to develop muscular dystrophy? Well, this has been a hard question to answer because many of the case reports that I came across were in infancy when the diagnosis was first made, and the patients were often lost to follow-up. And the second is that facial weakness can be very difficult to differentiate from weakness due to the multiple facial reconstructive surgeries these individuals undergo. And then the other question is, do patients with muscular dystrophy then have subtle defects in smell or reproduction? But before I answer those questions, let me tell you a bit more about SMCHD1 biology and how it causes FSHD2. So SMCHD1 stands for structural maintenance of chromosomes, flexible hinge domain containing protein 1, and it serves as a master epigenetic repressor, so it turns other genes off during development. It plays major roles in X chromosome inactivation, and it silences a subset of autosomal genes, particularly those that are imprinted and genes hidden within repetitive DNA sequences, especially a repeat called the D4Z4 microarray, which I'll tell you more about. And it does this through stabilizing DNA methylation, repressive histone marks, and non-coding RNAs, and also by contributing to higher order chromosomal structure. In FSHD2, as I showed you, some of the mutations are null, and some are missense, but it's thought to represent a loss of function of the protein. So with loss of SMCHD1 repressive activity at one of those specific repeats on chromosome 4 leads to the ectopic expression of a transcription factor called DUX4 or double homeobox 4. So very early in development, DUX4 is expressed transiently and plays an important role in something called zygotic genome activation. This is when the maternal mRNAs and protein are cleared and the zygote's genome is first activated. But then during development, SMCHD1 binds to this region, it becomes methylated, and DUX4 is turned off. And for reasons that are not entirely clear, in muscle cells in FSHD2, SMCHD1 loss of function leads to hypomethylation of the region. DUX4 is again turned on, leading to muscle cell apoptosis. So going back to these questions of, is this one disease or two? Well, we've studied a cohort of patients with FSHD2 in collaboration with some Dutch investigators, and we saw no craniofacial, olfactory, or reproductive phenotypes. We also studied a cohort of Irania patients at the NIH Clinical Center in collaboration with colleagues in NINDS who performed a neuromuscular exam, muscle ultrasound, and muscle MRI, and we could identify no neuromuscular defects. So does that mean Irania patients don't express any DUX4? Well, that's also been difficult to answer because even in FSHD2 muscle biopsies, DUX4 is only present at very low levels at both the mRNA and protein level. But there's two potential workarounds. The first is that one can treat patient fibroblasts with two drugs, decitabine and trichostatin A, and these relieve the epigenetic repression at multiple loci. So one can ask whether the stability of repression of DUX4 is different in patients with FSHD2, Irania, and controls. And so when we did this, this is showing DUX4 mRNA. You can see that some of the Irania patients, this HDF is human dermal fibroblasts, some of the patients express DUX4, but it's at much lower levels than what we see in FSHD2 patients. The second workaround is to differentiate myotubes that you obtain from muscle biopsies myoblasts into myotubes, which is known to increase DUX4 expression. And so this is showing relative expression of DUX4 mRNA in the undifferentiated myoblasts and at day 3 and day 6 of differentiation towards a myotube lineage. And you can again see that there is much higher expression of DUX4 in the patients with FSHD2 compared to Irania. So I think this at least tells us that in this cellular system, there is very differential control of DUX4 expression in these two conditions. And Irania patients may actually be protected from FSHD2 through some mechanism or developmental switch. So what do we know so far? Irania appears to be an extreme form of Kallmann syndrome, but we see no genetic overlap. There's very strong statistical evidence that missense mutations in SMCHD1 are the primary genetic driver of this condition. Both loss of function and missense mutations in SMCHD1 also cause FSHD2 via the DUX4 muscle toxin, but Irania and FSHD2 appear to be mutually exclusive conditions. Now, before we jump on the SMCHD1 bandwagon, however, I need to tell you a little bit more about these patients and to tell you that it really is a much more complicated genetic architecture. So the truth is that not every patient with a missense mutation in SMCHD1 has an abnormal phenotype. And this is called something in genetics called incomplete penetrance. So in this family, the mother has completely normal smell, normal nose, reproductive function, but carries a heterozygous missense mutation in the gene. She passed that variant to one child, a daughter, who has hypogonadism and complete Irania, and to a son who also has hypogonadism and nasal hypoplasia. So this pedigree also demonstrates something called variable expressivity. So these individuals have the same mutation, yet they exhibit differences in disease severity. And here's another pedigree showing that, that same pattern. So here's the father, where he has a variant and has nasal hypoplasia, passes it to one daughter shown on the far right who has Irania and hypogonadism, and to a second daughter who has nasal hypoplasia. So in summary, in Irania, we see variable expressivity, incomplete penetrance, and something called pleiotropy, or individuals with the same mutation exhibit completely distinct diseases. And what this tells us is that mutations in SMCHD1 are necessary but not sufficient to cause either condition. And it demonstrates that there must be a role for either genetic or environmental modifiers or for stochasticity during development. So how are we going to identify these modifiers? Well, what we really need to do is to open a wet lab. Well, this is a problem because I haven't held a pipette since my college days. But thanks to Daryl Zeldin and the NAEHS leadership, I was given the resources and personnel to open a wet lab several years ago. Because what I really needed to do as the next step is to model these missense mutations in human cells. And the two questions that really were the most important to answer at the outset were, what do these missense mutations due to SMCHD1 function? Do they make the protein less active, more active, or give it a novel function? And second, what cell type should we study? So we could start out with an embryonic cell type, but it's unlikely that that's what this mutation affects, or we would see more systemic effects and not effects just limited to the nose, eyes, and reproductive system. So some of the strongest evidence we had that these behave in a loss of function manner came from our patients. So we looked at the methylation level of the DUX4 promoter in that chromosome 4D4Z4 region, because in patients with FSHD2, part of the clinical diagnostic criteria are hypomethylation, so less than 25% methylation at that locus. And when we looked at our Irania patients with an SMCHD1 mutation, we saw that the majority of them also had hypomethylation at that locus, whereas the unaffected family members did not. And what about the cell type? Well, we needed to find a cell that could explain that clinical triad of Irania, microophthalmia, and hypogonadism. So we looked back to the embryonic neuroectoderm. So this is showing you the developing neural plate, which will eventually invaginate and form the neural tube to give rise to the brain and spinal cord. But there's two very interesting cell populations right at the neural plate border, the neural crest cells and the cranial placoid cells. Now, the neural crest cells migrate throughout the body, giving rise to parts of the heart, the enteric ganglia, melanocytes, but the cranial subdivision of the neural crest cells give rise to the facial skeleton, periocular mesenchyme, and olfactory nerve and sheathing cells. We've already talked about the olfactory placoid, which gives rise to olfactory neurons, GnRH neurons, and also releases growth factors that induce the formation of the nasal skeleton. And these two cell types really interact across development. So if we go back to our diagram of the developing olfactory placoid, here are the olfactory neurons extending their axons through the cribriform plate. But it's the cranial neural crest cells that give rise to these cells in green called olfactory and sheathing cells. And these are critical for the integrity of the olfactory neurons. So it really could be either of these two cell types. And in fact, when we look at animal models, we see that SMCHD1 is expressed in both cell types. So this top shows XGAL staining of mouse embryos expressing LAC-Z from the SMCHD1 locus. And you can see deep staining at E9.5 in the area of the nasal placoid. Below shows SMCHD1 expression in Xenopus, and you can see expression in the eyes, in the center panel in the nasal placoid, and then in the 4 migratory streams of neural crest cells. So now we have our wet lab. We have patients with Irania and FSHD2 as well as controls. We were able to obtain skin biopsies to get fibroblasts, derive iPS cells. And we also, again, because we're assuming this is a loss of function mechanism, we used the H9 human embryonic cell line and knocked out the SMCHD1 gene. And this is really helpful because these cells have an isogenic or the same genetic background, which can take away a lot of the variability. So I'll just tell you that we first tried to make neural crest cells, and we saw no abnormalities in proliferation or migration. And so we next followed a 11-day cranial placoid differentiation protocol that involves dual inhibition of TGF-beta and BMP. And we saw something very interesting right off the bat. So this is showing 2 lines of our wild type cells, and these are our knockout cells. And this is showing crystal violet staining, where the purple indicates viable cells and the white indicates cell death. And so you can see that around day 3 to 4 of differentiation, the knockout cells began to die. And why did they die? Well, it's very interesting that we saw right around the same time, this is showing DUX4 mRNA expression started to go up around day 2 or 3. And at the protein level, you can see in the SMCHD1 knockout cells, intense staining for DUX4 at day 4 and 5. We also did a time course RNA-seq experiment, and this is a volcano plot showing that many genes in red were upregulated and many were downregulated, shown in green. But the ones that have the names listed are all known DUX4 targets. And I'll just point out TRIM43, which will come up again later in the talk. So next, to demonstrate that this phenotype was specific to the loss of SMCHD1, we knocked back in SMCHD1, shown here with the KI. You see introduction of SMCHD1, which led to loss of DUX4 expression. And this is showing the plaquoed cells at day 11, and the knock-in cells, you can see recovery of cell viability. We also confirmed DUX4 pathogenicity using 2 different methods, siRNA and a pharmacological approach. So this is showing this, these 2 siRNAs reduced DUX4 expression. And here is showing, again, plaquoed cells on day 11, showing some recovery of plaquoed cell viability. And the second approach, as I mentioned, we used a drug called IP300W, which is a P300 CREB binding protein inhibitor. And with this, with treatment with the drugs, we saw loss of DUX4 expression and again, recovery of our plaquoed cells. So what about the patient-derived cranial plaquoed cells? Well, they behaved very similar to the knockout cells. So this is showing those crystal violet staining again of 3 wild-type plaquoed cells at day 11 of differentiation. And here are 2 knockout cells. And at the bottom are showing 2 healthy controls showing normal plaquoed cell development, 3 lines from Irania and 2 from FSHD2, again showing variable degrees of cell death. If we look at TRIM43 as our readout of DUX4, because it's much easier to measure, you can see again robust induction of TRIM43 in the Irania and FSHD2 lines, but not in the healthy controls. And I think what's very interesting here is that we see the same variability in phenotypes at the cellular level that we saw in the pedigrees that I showed you. And so this again points to a genetic modifier, and here we can blame it on DUX4. So something is modifying the expression of DUX4 differently in the Irania and FSHD2 cells. So how are we going to find that? Well, we proposed to do a genome-wide siRNA screen. And so we came up with this construct where DUX4 binding to its responsive element will trigger nanoluciferase activity. And here's our negative control with no DUX4 responsive element. So we transfected our plaquoed cells at day 3 with these constructs, and we can see that we see increased nanoluciferase activity indicating DUX4 binding in our knockout cells and none in the wild type cells. So what we're looking for in this screen are genes which, when knocked down, lead to a decrease in the signal in the knockout cells or an increase in the signal in the wild type cells. And so to do this in a high throughput manner, we're collaborating with NCATS, which has plate readers that can detect the luminescence. But while we're waiting for the results of that experiment, we've begun to think about possible environmental modifiers. And this paper recently caught our attention, showing that TRIM43 is part, during herpes virus infection, TRIM43 becomes upregulated as part of a germline specific transcriptional program, again, mediated by the transcription factor DUX4. So we asked whether herpes virus infection of our patient IPS cells could similarly induce DUX4 and TRIM43. So we infected the IPS cells, controls FSHD2 and Irania cells with HSV1. And here you can see increased expression of DUX4 after, this is pre-infection on the left, post-infection on the right. You can see increased DUX4 expression in the presence of the viral protein ICP0 shown at the bottom. And here again is our favorite DUX4 target, TRIM43. This is showing pre-infection on the left and post-infection on the right, showing induction of TRIM43 in FSHD2 and Irania IPS cells. So putting this all together, we started out with just a single patient with Irania. We're able to form an international consortium to build a cohort of patients. And through next generation sequencing, we're able to pin this on the gene SMCHD1. However, the variable phenotypes we observe in our patients suggested that there must be a genetic or environmental modifier. And functional modeling in the lab suggests that plaque code cells are the primary cell that's affected in Irania, and that cell death is due to the DUX4 toxin, just like DUX4 is a toxin in muscle cells in FSHD2. And we also have some clues that HSV1 exposure may be involved. And there are a number of questions that still need to be answered. What is special about muscle and plaque code cells that allows for DUX4 to be de-repressed upon loss of SMCHD1 activity? And why are there no individuals with both disorders? Is there some sort of developmental switch that protects one from developing the other? And are there other environmental contributors that we should be investigating? I want to thank the many people who helped with the initial genetic discovery, as well as colleagues at NIEHS and at the NIH Clinical Center who helped with the deep phenotyping of these patients. And in particular, Karu Inoue in the lab, who did all the experiments in the plaque code and IPS cells. And then a big thanks to my mentors at NIEHS and Harvard. Thanks for your attention.
So I guess I can moderate. Why don't you stop sharing your screen, Natalie? If there are any questions for Natalie, maybe I can start out. So would the work that you've done be possible anywhere other than the NIH Intramural Program?
Probably not.
That's the
right answer.
I think that was the right answer, Natalie.
So scientific questions for Natalie.
Natalie, I have a question. Thank you very much. Very interesting work and important work. You mentioned early in the presentation that there's a role for the SMCHD1 gene in as being imprinted. So how does that play into some of the cases you see and the effects of the protein mutation in either the father or mother? Yeah.
I should mention that most of the functional studies have been done in the mouse. So we actually don't have evidence of defects in X chromosome inactivation or imprinting defects in our patients. And in fact, we see males to female and females are equally affected.
Other questions for Natalie? Or are people just tired because it's 4:05 in the afternoon and we've been going for 6 hours straight?
Quite honestly, I think I'm just speaking for myself. I'm blown away by this. It's just a beautiful piece of work and incredibly meticulous. And then still at the end, there's a little bit of a mystery, which was disappointing, but fun because it means that there's more that can be done to elucidate what's going on here. But congratulations for that. That was really, really beautifully presented as well as done. I really appreciate it.
Thank you very much.
Natalie, it's really impressive to see how much progress you've made in the relatively short time you've been at NIEHS. Very well done.
Thank you, Rick. It's been a lot of fun.
So maybe I'll turn things.
I would echo. Oh, sorry.
It's okay, Dar.
Go ahead, Irvin.
So I would echo what Lynn said. It's what a fascinating story, and how you sort of took it from in all directions. I have two questions. One is, I noticed on the map, and I don't know what kind of bias there might have been in relation to how the cases fell out geographically, internationally, but it looked like there was really a big cluster in the Scandinavian countries. And I wonder if, does that seem to match up? And I don't know if you've looked at this, with the genetics in the cases that, maybe some sort of.
Yeah, no, I
origin
think I
that
need
way?
to update the map too.
Okay.
But yeah, I think that was just more of a convenience. That's where the main collaborators were, but we've since identified, they're on every continent, and we have patients in Palestine, in Colombia, in Japan, Africa, all over. And I think there's probably one in every US state. It's rare, but it's not as rare as it might appear from the literature.
Okay. And then I may have lost you, but was the environmental factor that you were talking about, was the herpes component of it? Okay. Yeah. Thanks.
Maybe I'll turn things back over to you, Pat.
Okay, thank you, Daryl, and thanks, Natalie. That was a great talk. Appreciate your
Thank you.
joining us today. Rick, I will go ahead and introduce our next person, or would you like to?
I'm set to introduce Joel, if that's okay.
Let's do it.
Okay, terrific. Well, in the interest of time, we're running a few minutes over, so I'm going to be brief in my introduction. I just want to say that it's truly a pleasure to introduce Dr. Joel Kaufman, who is a professor in the departments of Environmental and Occupational Health Sciences and Medicine and Epidemiology at the University of Washington. So he's had just a number of remarkable accomplishments, including being elected to the National Academy of Medicine back in 2020. But of course, we think that one of his most noteworthy accomplishments was being selected in a competitive search to take on the additional responsibilities to be the editor-in-chief of EHP, and this was back in 2019. Under Joel's leadership, there have been quite a number of just remarkable developments of the journal. So we thought it would be good for Joel to give a presentation and to update our council and other members of the environmental health sciences community and the things that have been happening at EHP. So Joel, without further ado, let me turn the virtual platform over to you. Oh, Joel, are you there?
Okay, I'm there now. How's that?
Okay, there you are. Great.
Okay. Thank you for that introduction, Rick. And it's a pleasure to present to the council. I recognize people are running a little bit late today, so this isn't intended to be a very long presentation, really just an update on a few things and to give you a few highlights of things that are going on now at the journal and things that are going on in that we're projecting over the next period of time. Let's see. People will recall the journal. This has been a transition for the journal in that prior editors of the journal had been NAEHS staff scientists up until 2 years ago, when I was named as the first outside person as editor-in-chief, and we thought that it would be a bit of a challenge to figure out how to have a remote editor during this as a transition for the journal. And it turned out, of course, that not only was there a remote editor, at about the same time, all of the journal went remote in operations due to situations beyond our control. So it's really put me on a level playing field with the rest of the staff in terms of being at remote along with everybody else. And nobody's really been back in the office now over the last couple of years, of course. But I want to talk a little bit about how this has all worked and what we've been up to. Maybe things haven't all been as easy to do because of the pandemic, but it's been a period of fairly rapid change at the journal. We've changed a fair number of the processes that have that the journal uses both for editorial workflow and other procedures. We had a strategic planning process, which I'll very briefly touch on today. And we've launched a few new initiatives at the journal, and I'm looking forward today to getting some input on a couple of the initiatives that we're have launched or will be launching soon. The strategic planning summit that we had through an open space technology, virtual open space technology, now a little more than a year ago, was extremely well attended. We had almost 100 stakeholders who participated in this set of meetings and got lots of good input and ideas to help us think about how to prioritize activities at the journal. It's a special journal, I think, for a lot of people in the environmental health sciences. And of course, with support from the NIH, we are privileged in that the journal can both be a fully open access journal to anybody anywhere in the globe without any kind of a paywall, and also is able to operate without any publication charges for contributors. So we're in a good place, and our strategic planning allowed us to think about how to prioritize a variety of things. And we, in the course of the strategic planning exercises that followed the summit, we did revise our vision and our mission, and they're shown here on this slide. I'm not going to read them to you today, but we really want to be if not the leading journal in the field, and really work to have high impact content and be the journal where people want to publish their best work, and the journal where people in the environmental health sciences know they can turn for reliable information and cutting edge information on the science of environmental health, as well as policy implications of it. So our first goal that we have as a journal is to attract and publish novel, high quality research. And one of the aspects that we want to feature is the fact that we're taking contributions from all disciplines that comprise the environmental health sciences. And that means, many disciplines, not just my own sort of background in both clinical and epidemiological research, but also the toxicological sciences, exposure sciences, and a variety of other allied fields, including methodologies in biostatistics and fundamental science and exposure sciences. So there are lots of ways that we can address these issues. Many of our, externally, of course, many people focus on the impact factor, and while it's not something that we want to focus on as our main metric, we really want to be a journal that people trust and go to and want to publish their work in. We have seen our impact factor rise steadily over the last few years, as shown here on the screen. But what I want to focus more on is what we've done to revamp our editorial processes at the journal, which have really been substantially reworked over the last 2 years, and now incorporate a set of 3 deputy editors who are closely involved, external academic leaders who are involved in helping with the triage and the decision making about every paper that comes through the journal now. And those deputy editors, not shown here, are Paige Lawrence from the University of Rochester, who is a senior established toxicologist, and Manolis Kojovinas, who's a established environmental epidemiologist, and Dana Barr from Emory University, who is a renowned exposure science expert. And between the 3 of them, we really have elevated the ability to get sort of a quick external read on impact, and also provide outreach and be sort of an ambassador to those communities of scientists to help them know that there's somebody at the journal looking out for the interests, at least of the areas that they're focused on. Of course, we also have a board of associate editors that are experts in sort of all of the disciplines that we see come into the journal. And we have, as I indicated, reworked our workflows in an effort to try to speed things at the journal. This slide is just to show you that we do get publications from across the range of disciplines. We often have heard critique, for example, that we're just the journal of environmental epidemiology. And while epidemiology, shown here in the light blue, continues to be the biggest source of submissions that we get, and the single largest area of accepted papers, it's actually no more. We actually get a slightly smaller proportion of the submissions accepted, and you'll see that some other topics like toxicology actually have a larger proportion of the papers ending up being accepted by the journal when they come in. So we just want to make sure that folks are sending in their best stuff so that we can evaluate it and publish it in the journal. And a big focus has been on timeliness at the journal. Now, again, the last couple of years have been challenging for the review processes in general due to the pandemic. A lot of reviewers are saying no to, more reviewers saying no to requests, and it's harder to get reviewers for papers. But you can see we have a steady improvement in our timeliness of our decisions, including time to first decision and time to final decision of a paper. I think most would agree that although we've dramatically improved our speed to sort of that initial decision, especially for those papers that are not going to get sent out for full review, we are not a fast journal in terms of publishing work that's eventually going to be accepted. I think we're probably never going to be the fastest journal in that regard, but we are very proud of the product that we publish and the quality of the papers that come out in the journal, and we do put a premium on the rigor of our review process and the care that goes into making sure that we can be proud of everything that we publish. There have been several other things we've undertaken over the last few years with regard to what we now characterize as our second goal, which is communicating findings and knowledge to a broad array of stakeholders. And we've tried to do things beyond the publishing of original research, which is obviously most of what we do, in addition to the news content that we publish. And we've had several curated collections to highlight work that we do. We've published, for example, our environmental racism collection now in 2021, which was an update of work that we had done on the topic in the past. We've done a recent developmental toxicology collection. And then the other thing that we've done is we've rolled out and changed our article types, and we have more new article types coming. Most notable thus far has been the reintroduction of a letters section, including both a letters to the editor section as well as a research letter, which is really a kind of brief communication, a kind of short piece that people can share for more rapid publication of content that doesn't merit a full-length research paper, so the research letters. And the other thing we've begun is invited perspectives, which are editorial content related to either research papers or commentaries that we publish to provide an expert perspective on something that we're publishing. And those have been appearing over the last several months and have proven to be very popular new content in the journal, and a way to have more people participating, get more leaders in and sharing their views about what we're publishing. So many of you may not be aware, but this year will be the 50th anniversary of the journal, and I don't have time to go into the history of the journal here. The journal has evolved and developed a lot over this period of time, from being not only one of the first environmental health sciences journals, but also being a journal that was largely publishing work that was NAEHS symposia products initially, and conference proceedings, to a full-fledged peer-reviewed journal, scholarly journal, and a leader in the field. And to celebrate the 50 years, starting in the spring of this year, we'll be hosting a series of things on our website, including an interactive timeline of the history of the journal, video interviews with past editors-in-chief, and a series of editorials about what we've accomplished and what we're looking to do over the years to come. So look forward to that coming up later in the year. The other goal that we have developed is really around how the journal can play a role in fostering the development of an innovative, diverse, international community of contributors in the environmental health sciences. And there are two major pieces to this that I want to focus on. One is the Early Career Researcher Initiative that we launched last year, and the other is the re-envisioning of our international program, which will be going on, which has been going on and will continue through this year with some exciting developments to come. And I'll tell you a little bit more about that. So we developed last year an Early Career Researcher Initiative, which involved a few things, including the recruitment of an advisory panel of early career researchers who have been working with us closely to develop activities and programs that we can be rolling out. We have developed a mentored review program to make it encouraged for established investigators to work with early career investigators in learning how to review papers. We've been working to develop a pool of early career researchers who can become part of the reviewer population for the journal. We've been giving mentored writing opportunities for early career researchers. We've published a peer review resource center on the website, and we've relaunched the Tuesday Tips program on social media. So I want to talk a little bit about our international program, which is in a state of transition right now. But in order to refocus our activity on really improving scholarly publishing capabilities in low and middle income countries, we are focusing and considering focusing, and probably will launch during the course of this year a sister journal to EHP. It's currently in the on the drawing board, but it's moving ahead. We can't make any kind of a public announcement of this for some time to come, but we are hoping to have a journal which will be published work from low and middle income countries, edited by editors from low and middle income countries with to meet the needs of those countries and to build a community of environmental health science scholars in resource poor environments around the world. So that's a really quick overview. Given the time, I know you guys are, your day is about over. So I thought I would just take questions or comments from people and see if you have questions for me or thoughts about EHP and what we need to do. Thank you.
Well, that was terrific. Really a nice summary of lots of things that have happened at EHP over the last couple of years. Looks like Phil Bourne has a question. You have your hand up, Phil?
I do, yeah. Thanks very much for that. I confess that this is not particularly my area, so I wasn't overly familiar with the journal, so I apologize for that. I really enjoyed your presentation. I was also thought about, and I don't want to necessarily get into this, I'd love to talk to you offline about it, but the political ramifications of all of this, when I think about the notion that the NIH is running a journal, adverse and how that plays with commercial publishers, but that's a separate issue. But perhaps the important thing here is what advantage, how can you talked about the content and the goals of the journal, and it's all great to me. I was thinking more about the context, the opportunity to push the envelope in scholarly publishing in the way that the NIH is trying to push various aspects. So I was thinking particularly of data. So with the new data sharing policy coming next year, I'm just wondering whether there are ways that the journal can actually be the sort of flagship for moving scholarly communication forward in the way that NIH is pushing from behind, and the journal could be out in front. Am I making sense?
Yeah, and I think it's a really important point. And I think one of the issues that that we'll be wrestling with, I think, is as data sharing and data access initiatives move forward at, across the NIH, how we can be a model in showing the best practices for data sharing and so forth, and whether that means working with data repositories elsewhere at NIH to have a sharing portal, for example, for data from things that we publish. Clearly, one thing that we make, immediately, easy for work is that everything we publish is open access and everything is, not behind a firewall. But I think the expectations for sharing of data are going to be increased, and we'll need to be moving, lockstep with that and trying to stay ahead of the game. So I appreciate that comment. Couple more hands up. Do you want to call on them, Rick, or should I?
Yeah, sure. So Lynn, it looks like I think you were next. Do you have a comment or question?
Yeah, I do. Hi, Joel. It's great to see you. I also, have some interest in the data sharing area, and particularly, it seems to me that maybe even this council might be able to assist you with that at some point. I do think that this is a highly complex issue when it comes to environmental health sciences as compared to even genetic data and other data where we have clearer data taxonomies, and there's just a lot more clarity also about some of the issues around data privacy and individual privacy. I know that a lot of people in the environmental health community are quite concerned about the issues around human subjects and privacy, whether it's because they're working with Indian tribes or they're working with children, or the kinds of consents that we've had with our study subjects. And so I think this would be a great group here. I won't be on this forever, but to help to inform that. I think as another kind of piece of this is that, increasingly there's a lot of very, very big data that are involved in your publications, and how you share that, the larger data, the models, where sometimes not every institution can even run a lot of these models because you have to have very high performance computing capacity to do that. And what your thoughts are about that. But I just want to congratulate you. I've seen how Lancet has spun off some other journals, and that's been very effective in creating more channels for people to publish. And I do think that we badly need that in the environmental health field globally. It can keep up the quality of the journal. And because we have seen a tremendous growth and diversification across the world in environmental science, and it's very difficult for many to find outstanding channels to publish their work. Great work. So thank you for doing that.
There was a lot of comments there. I'm not sure that was a specific question around how we're going to do data sharing and so forth, but it's obviously something we're going to have to be very thoughtful about and make sure we're not requiring things that aren't fair to require either. So that's
and just more of a comment to that this group or others, but I think this is a good one, could help help with that
happy to
as you develop your strategy. Yeah.
I see we're about out of time here, but I see Katrina's had her hand up. So.
One more question or comment from Katrina, and then we're going to have to wind things up. Katrina?
I'll try to make it super concise. Thank you for that presentation and all the great stuff the journal is doing. I really think this is such an important forum for the developing the field. And I'd like to take what Phil and Lynn said and run it to the other side of the research spectrum. One of the things in partnerships for environmental public health that we've been talking about is what is the meaning of innovation when you're talking about community engaged work and partnerships with communities. And that's so important to NIEHS. I wonder as a journal, how you are thinking about innovation in terms of impact in community engaged research.
I think we're, the short answer is that we're interested in it. The long answer is that we can only publish things that get submitted. So, you know, we try, the question is, how do we prioritize things that come in? And how do we encourage good work to come in? And how do we get the best work in this, you know, exciting, innovative area of community engaged research to be structured in a way that it, you know, will be, do well in the review process and get through the journal. I mean, there's sort of all of that goes on, but we've, you know, as part of an initiative and early in our days, we added 2 new associate editors, both who have an interest in community engaged research and in social science interfacing with environmental health sciences research, which has been you know, has allowed us to think more creatively about how that, how innovation is important. And we're going to continue to emphasize that we're interested in publishing it in terms of what is innovation. You know, I think that's, you know, a little bit in the, you know, in the eyes of the storyteller in the paper to explain to us why it's innovative and why we need to be interested in it. And then we can, you know, put it out to readers and see if they think it's innovative. I don't know what more I can say.
So Phil Bourne, I just noticed that you put a very important comment in the chat box. Do you want to unmute and just share that with the group before we adjourn?
Yeah, sure. I partly this is, I should say, this is not really a fair comment because in 2005, I started one of the PLOS journals and I wrote an editorial about the linkage between the knowledge that's in a paper, the data that from which that knowledge was derived, as well as the methods, and how they should be much more integrated. It's going to be my epitaph, I think, because we're still a long way from having, we actually have that in different contexts, like Jupyter Notebooks. But I just think that with the NIH having a journal, it's an opportunity to push various types of technologies in the scholarly communication space and be a real exemplar. So it's really just building on what I was saying before.
And it sounds like we can probably benefit by picking your brain in another forum and trying to get a little more thought from you.
I love talking about this stuff. I'd be happy to, to chat with you anytime. I'll reach out to you.
Great.
We've been privately chatting with Phil about a visit once we can do that. Remember actually gathering in person? We might be able to do that at some point in the near term. And then Joel, we actually may have you in for that visit from the University of Washington. So I think we better wind things up today. We're running just a few minutes over. So thanks to everyone who gave a spectacular presentation today. I found it to be really quite exciting. So we will adjourn now, and I look forward to seeing all of you tomorrow morning. So have a good evening, everyone.